We are going to start our next session, everyone. So as you can, grab a coffee and grab a seat. So welcome back, everyone, and a big Texas hello to those who are joining us online. Thank you for being here, and thank you for being here in person here in Dallas. Our virtual and in-person attendees, remember you can submit with the QR code, the question and answer box at any time. Please do as you're thinking about those questions. Our next session is going to be led by the masterful and brilliant Dr. Carl Gay. Dr. Gay is an associate professor at the University of Texas MD Anderson Cancer Center. He focuses on both clinical and translational research in small cell lung cancer. His work is helping to drive forward new insights into the biology of this disease and how we can target it. So please join me in welcoming Dr. Carl Gay for exploring genetics and biology, blazing new trails in small cell lung cancer. Thank you and welcome, Dr. Gay.
Thank you, Dr. Shields, for the kind introduction to longevity, to Misty, to everyone for the opportunity to be here today and to talk to you. This is obviously a topic that's very near and dear to my heart. And thank you to all in the audience who are in attendance, everybody that's online. My goal is to make this a little bit conversational if we can. And so I have a few slides that I've prepared that go into sort of the next steps beyond Dr. Singh's talk. I think Eric did an amazing job of outlining where we are right now with small cell lung cancer, and there's clearly been some amazing strides in recent years, but the one commonality, unfortunate commonality with pretty much all of that data is that it remains predominantly, if not entirely, one size fits all in its design. So every patient with small cell lung cancer is being treated essentially the same way, identically, even though we know that there's variety across the disease. And so I'll talk a little bit about that variety, a bit about some of the new drugs on the horizon and how they may be able to exploit that variety as a vulnerability in ways similar to what we do with non-small cell lung cancer. And so I think there are two obvious ways that we can improve the lives and the lifespans of patients with small cell lung cancer. We can either make better drugs or we can improve our biomarkers and our patient selection so that we're making the most out of the drugs we have. And unsurprisingly, I think we need to do both of these things in order to really move the needle. Let's start with improving our drugs and talk about some of the ways that the therapy for small cell lung cancer patients might change in the near future. And so the history of small cell lung cancer is unfortunately quite a boring one until very recently. And so when I first started giving talks about small cell lung cancer and writing grants about small cell lung cancer, I used to always use as an intro that there had been no change in small cell lung cancer therapy in over 30 years. And that was unfortunately true until very, very recently, right? That most of the improvements that we had seen, new therapies, successful trials, were in the late 70s to the mid 80s. And that's a uniquely bleak period across the cancer landscape. Fortunately, as you've heard from Dr. Singhi, that has changed. And so if you look at these timelines on here, you can see that the majority of the circles, which indicate new successful trials or sort of imminent trials, are backloaded, right? So we had these decades where there was nothing going on and just a total dearth of new options. And now we have immunotherapy and lurbinectedin plus immunotherapy. And we have tarlatamab and all of these new things. We have immunotherapy in the limited stage setting, all of the things that Dr. Singhi talked about. And so there's been a real explosion of new therapies, new approaches in this disease, and even totally new mechanisms of action of our drugs, right? You heard about tarlatamab, this T-cell engager, the super glue analogy I'm going to steal from Eric and Misty, because I'm always test driving new analogies for tarlatamab in the clinic, so I'll give that one a run too. So suffice it to say, we have more tools for small cell lung cancer than we've ever had. But as I said, they're all still one size fits all, even though the dirty secret of it all is that we know they're not one size fits all, right? We're using them in a one size fits all fashion because we don't have any choice, but we know that we can do better than that. And so one of the ways that small cell lung cancer therapy is changing, and this is not specific to small cell lung cancer, there's some overlap with what's happening in non-small cell lung cancer and other solid tumors, even in liquid tumors, but attacking what I'm going to call the surfaceome. And that's a really jargony term. Basically what that means is proteins or molecules that are expressed on the surface of a cancer cell. So cancer cells are really abnormal. They do not look like say a healthy lung cell that might be adjacent to them. And this is what makes them tick. And you can imagine that you can use some of this abnormality as a potential vulnerability if you exploit it in the correct way. And so a few of the ways that we're exploiting this surfaceome. So there are what we call monoclonal antibodies. Examples include drugs like atezolizumab, Tecentriq, or durvalumab, Imfinzi. Right? These are monoclonal antibodies against PD-L1, which tends to be expressed at the surface of cancer cells, although unfortunately not very often in small cell lung cancer. Getting sort of more sophisticated and more science fiction-y as we go, there are also antibody drug conjugates, or ADCs. This is one where I've been test driving a number of different analogies. A lot of people are using heat-seeking missile or smart bomb. I like Trojan horse, but I should solicit feedback at the end here. Basically, the way that this works is there is an antibody that is attracted to a protein that's expressed on the surface of the cancer cell. And when an antibody binds to the cancer cell may welcome it in, right? It sees it as potentially friend. And then hidden on the back of that antibody is a huge dose of chemotherapy, one that would be intolerable if given to all of the cells of your body equally. But so the antibody gets welcomed in, the bomb explodes once it's in there, and you can direct a big dose of chemotherapy to a very limited area. And so theoretically, this should deliver chemotherapy only to cells that express this protein. If you pick a protein that is sufficiently specific to the cancer cells, you can deliver chemotherapy only to the cancer cells and minimize the collateral damage. Another way that we can exploit the surfaceome is what we call CAR, or chimeric antigen receptor cells, usually CAR T cells. And so this is a more sophisticated, convoluted process where your immune cells are taken, the patient's immune cells, I should say, are taken from their body. They're genetically engineered to have only a one-track mind, where they become immune cells that only care about a single protein, and you design them to care about a protein that's on the surface of their cancer, and infuse those cells back into their body, and the cells should go nuts on the tumor and leave normal, healthy cells in the body alone. And so this is something that has been tested and has shown some success in small cell lung cancer patients in early phase, what we call phase 1 trials. It's proven to be safe and potentially effective, but still a lot of work to do. And then you heard a bit from Eric and with the superglue analogy on the T-cell engagers, right? So this is like tarlatamab and several other therapies, and so I won't go too much into that because he did such a nice job. And so these are the ways that therapies are changing. There are a number of these proteins that are expressed at the cell surface. You've heard a little bit about DLL3 or delta-like ligand 3. That's the target of tarlatamab. But there are others like B7H3, SEZ6, TROP2, and several more that are in the works. And I'll talk a little bit about those. So T-cell engagers, you've seen, I think, this exact figure actually in Dr. Singhi's talk, or something very similar to it. This is the superglue. This is forcing the T-cells that are already present in the body to face off with the cancer cells. So small cell lung cancer in particular is very good at excluding T-cells from what we call the tumor microenvironment. So the small cell lung cancer tumor is we often use the term immune desert. So the immune system essentially ignores it, or maybe the small cell lung cancer tumor is actively repelling it. I think there are debating theories on that. But the immune system ultimately is not doing much to help the patient control their cancer. This forces the issue. It forces a face-off between the T cell and the tumor cell. And if DLL3 is there and everything gets engaged, patients can have really beautiful, durable responses to these T cell engagers. And I think all of us have experience with patients that are well over a year out on tarlatamab and other drugs like this. Durability of responses that we could have never imagined, and depth of responses that we could never imagine. These are the ADCs, or antibody drug conjugates. So I talked a little bit about this. This was my Trojan horse analogy. So again, the ADCs, they bind to a surface marker. They sneak a bomb into the interior of the cancer cell. There's also a phenomenon that they call bystander effect, where if the bomb is sufficiently powerful, it may kill not only the tumor cell that ingested the antibody, but also the surrounding tumor cells. So there's a delicate balance there, right, of titrating that effect so that the bystander effect is confined to the tumor. This explosion that happens with ADCs can also potentially trigger an immune response. And so there might be a rationale to combining this with immunotherapies like durvalumab or atezolizumab, or even T-cell engagers, because it may provoke the immune system. It may make the T-cells finally stand up and say, hey, something is not right here. We should not be ignoring this enemy in plain sight. Some of the drugs that fit into this category are things like IDXD, or ifinatamab deruxtecan, if anybody has heard of that. Sacituzumab govitecan, which is, so those are against B7H3 and TROP2 specifically, and there are phase 3 studies that are already ongoing for testing those in small cell lung cancer patients who've had their cancers recur. The other one listed there, that just a alphabet soup of letters and numbers, is a drug that targets SEZ6, and that now has entered into phase 3 studies. And so these are not 5 years away, pie in the sky ideas, right? These are drugs that patients can access right now for small cell lung cancer if they meet the criteria for these studies. And so these are things that we've seen work already. I have multiple patients on the SEZ6 ADC that have done really, really well for a very long time, much longer than we would expect with something that's chemotherapy-based. So this is a little bit about where I think the field is going in terms of therapies, but one of the potential obstacles, and it's a good problem to have, I want to emphasize that, is if all of these drugs work very well, we still have difficult decisions to make, right? If I have 3 T-cell engagers and 4 antibody drug conjugates at my disposal, and they all have similar response rates, I may not have the luxury to test all of those in a particular patient, right? Small cell is a sufficiently aggressive disease that if 1 or 2 drugs in a row fails, I might not get a third chance. I might not get a plan C or a plan D. And so making sure that we put our best foot forward with patients, selecting the right patient for the right drug is going to become critical. As I mentioned, historically, we haven't had to do this. We have not had the luxury of doing this because we only had a limited number of therapies. And so to restrict a patient from any of them would be unethical. But if I have 3 different ADCs that all generate 60% response rates, that's great in a population. I want to know what ADC would work for the patient that is in front of me in my clinic that day, right? I want to be able to triage and prioritize the selection of the drug. So how might we do that? And so I think many of you are probably familiar with the fact that in non-small cell lung cancer, this is exactly what we do. So when I see a patient with non-small cell lung cancer in the clinic, I visualize this pie chart in my head. I describe this pie chart to them where I say, look, we know you have non-small cell lung cancer. That's not nearly enough to decide on your treatment. I need to know what the immune environment of your tumor is. I need to know what the driver or driver mutations of your tumor are in order to select a therapy. If you have an EGFR mutation, I should put you on an EGFR targeted therapy. That will only work if you have an EGFR mutation. So this has been the way that we've managed non-small cell lung cancer for well over a decade now, and it gets more and more sophisticated really every year. And so this is it's a relatively satisfying process, I think, as both patient and physician to select something that you know is personalized, that you can give a response rate. Dr. Singhi mentioned some of these 70, 80, 90% response rates if you have the right match of drug and driver. In small cell lung cancer, it's I think, not quite that simple, but that's just because we haven't gotten there yet, right? And so I think if we focus on the genetics of small cell lung cancer, the way that we do in non-small cell lung cancer, if we think about the driver mutations, which is a buzzword that you'll hear a lot, we're probably not going to get very far because small cell lung cancer almost uniformly has the exact same mutations in every patient. So almost every patient with small cell lung cancer has an alteration in a gene called TP53 and an alteration in a gene called RB1. There's a little bit of controversy in making that as a blanket statement, but that's generally true. They might have a slightly different mutation in each one of those, but the challenge is that those mutations are not drivers. They are usually brakes, and so it's more that the brake lines have been cut rather than the foot is on the pedal. And it's a lot more difficult to restore the brakes than it is to stop the gas pedal in a cancer. And so there are some promising drugs for TP53 on the horizon, but I think we're not going to get very far in the immediate future if we think about the genetics of small cell lung cancer. Instead, if we think about gene expression in small cell lung cancer, and so the aberrant genetics that underlie the disease result in genes being turned on or off that are atypical for lung cells, for neuroendocrine cells, for tough cells or basal cells, or wherever this cancer is originating from. And I think historically, it was understood that maybe all small cell lung cancer tumors were not alike. When they grew them in dishes, they saw that sometimes the cells stuck to the plates that we grow them in, and sometimes they float in these clumps. And people said, that's weird. Why don't they all do the same thing? Why would a different patient's cells adopt a different phenotype in a dish? But we know dishes aren't humans. We've learned that too many times in cancer research. And so there was always a hint that there was something more to the gene expression profile of small cell lung cancer patients than met the eye. And so what we have shown and others have shown is that small cell lung cancer, based on these gene expression patterns, can be subdivided into multiple subtypes. And there is some nuance, depending on who you hear give this talk, as to exactly how you delineate these subtypes. We delineated them into 4. 3 of them are defined by what we call a transcription factor. This is sort of a gene that regulates what other genes do. And we have little acronyms for each of them, so you can be a SCLC-A, so small cell lung cancer driven by ASCL1, or the A subtype, or N, or P. And then what we contributed also was that there was a small subset of patients with small cell lung cancer that didn't have this immune desert phenotype that I mentioned earlier. They actually had T cells. They actually expressed the targets of atezolizumab and durvalumab in the tumor, and we called these inflamed, or I subtypes. And so basically, if you took the small cell lung cancer pie, you could divide it up across these 4 subtypes. And essentially every patient fit into one of these subtypes. So we predicted that the patients that had the inflamed subtype might be the ones that are actually getting benefit from immunotherapy, right? We know from Dr. Singh's talk that with the addition of immunotherapy, there is a benefit. It is modest. There are 2 interpretations of that. One is that every patient with small cell lung cancer is getting a modest benefit, and it's all equal. The other is that some small group of patients is getting a tremendous benefit from small cell lung cancer, the tail of the curve that Dr. Singh flagged, this 10, 12% of patients that are alive at 5 years, and they're doing all the hard work in that overall survival curve, right? They're pulling the numbers up so that it gives the sense that everybody is benefiting. So when we went back and looked at some of this immunotherapy trials, we found that while no patients failed to benefit from immunotherapy. So there's no one who's being harmed by immunotherapy, to be clear. There was a clear signal that the patients that had this inflamed subtype, 15% or so of the total population, were getting much more benefit than others from immunotherapy. So that tells us 2 things. One, that we got to make sure those patients get immunotherapy, and we should think of ways to double down on exploiting the immune system in those patients. But 2, the other 3 subtypes that lack those features, we have to do better, right? They need something more to stimulate the immune system. And we know that the only way that we really get long-term survivors or remissions or cures in patients with metastatic disease is to engage the immune system. We know that from non-small cell. We know that from other places. And so, at that point, this was a test that we could do in the lab with extensive utilization of our computational biologists and biostatisticians and everything. It was not a practical test that we could offer to the public. And so it was something that was interesting, but of no use to a patient that asked questions about it, of no use to a physician that was treating the patients. So we now do have an actual test with a company called Boston Gene. So you may have heard of other companies like this, Foundation, Tempus, Caris, these companies that do biomarker testing for patients with cancer. So Boston Gene is one such company. And with them now, I can take a patient that I see in clinic with small cell lung cancer, or another number of other similar cancers to small cell lung cancer. I send them somewhere between 5 and 10 slides from the patient's biopsy, and they send me a report that looks like the far right of the screen, which tells me which subtype these patients would be assigned to. Now, in some cases, there's something I can do about that. In other cases, there's not. But it's useful to know, just like it was useful to know patients had KRAS mutations in non-small cell lung cancer, even though we didn't have KRAS mutations for many years thereafter. Right? And so I think this is really the first step towards precision oncology, personalized medicine in small cell lung cancer. We could finally subdivide patients into meaningful subgroups. And there's actually a clinical trial that's ongoing right now called the PRISM study through SWOG, formerly known as the Southwest Oncology Group. This is what's called a cooperative group that oversee trials at hundreds of centers, both big and large, or big and small, across the United States. And so with this study, the schema is a little bit complicated, and you don't need to memorize this for sure. But basically, patients who are newly diagnosed with small cell lung cancer, during that initial wave of chemotherapy that every patient gets, they have their tissue sent to Boston Gene. Boston Gene reports back in about a week that the patient has this subtype. And then based on that, and then one other test that I won't go into in the interest of time, patients are assigned to a particular therapy option in what we call the maintenance setting. So after they finished their initial 3 months of chemotherapy, when ordinarily they might be getting immunotherapy or immunotherapy plus lurbinectedin, these patients can be assigned to something that we think is a vulnerability of their specific subtype. And so for example, for the inflamed group, we're doubling down, just like I said, and we're targeting not only T cells, but something called natural killer cells in the inflamed group, because we think that having more immune cells caring about the tumor and fighting the tumor would be better. And so this is really one of the first efforts to personalize therapy for small cell lung cancer patients, where there is an option for everyone. There have been biomarker-driven tests before. Those were have and have-nots, right? You either had the biomarker or you didn't. If you had it, you could go in. If you didn't, you couldn't. Here, there's something for everyone, as long as they have enough tissue and the results are clear. And so this is very gratifying to see, and I think we'll continue to see efforts around these subtypes or variations on these subtypes that integrate some of those new therapies that I talked about, antibody drug conjugates, T-cell engagers. At the time we were designing this study, it takes a long time to make a study. This will prove that, right? Those drugs were not proven. They were twinkles in their mother's eye, right? And so by the time you get the study open, you realize, hey, now there may even be better ways to do this in the future, and you start working on version 2, version 3 of these studies. Other ways to think about discovery of biomarkers, I'll call out the work of Dr. Shields here, right? And so thinking about patients that you've had in clinic that have what we call exceptional responses. And so this is what she did with patients who had received lurbinectedin, right? That she had some patients in clinic who, all have patients that have benefited from lurbinectedin, but I think all of us also have patients that have really, really benefited, right? That almost inexplicably are on for 1 year, 2 years, et cetera. And you think, what is different about this patient that allowed them to continue to respond in a way that my other patient may have only responded for 3 or 4 months? And so they analyzed some of these cases of patients that had responded really, really well. They found a protein called ISL1 was more highly expressed in the patients that responded better. And so one could theoretically think of ways to triage patients to different therapies on the basis of these tests, right? You have some decisions to make in small cell lung cancer. Do I want to give tarlatamab? Do I want to give lurbinectedin, for example, in a patient that has relapsed disease? Well, knowing what their DLL3 status is for tarlatamab or their ISL1 status is for lurbinectedin might help you to make that decision. In an all-comers population, like Dr. Singhi showed, tarlatamab was better than lurbinectedin, but that does not imply that that's true for every single patient, right? There may be room to, osimertinib, for example, in EGFR mutant non-small cell lung cancer patients is really excellent, right? It does great and been very difficult to beat. But if you did that in an all-comers population, you would see some signal of that, but there'd be lots of patients in there that did not respond to it because they don't even have the right mutation. And so I think thinking about small cell lung cancer in that way is really prudent for us as investigators. But really what we need to do is to combine these things, right? We need better drugs and better biomarkers. We see this wave of drugs coming, and we need to be prepared to decide who should get which drug. And so I think we and others are already trying to do this. So when we think about DLL3, the target I mentioned several times, the target of tarlatamab, at our institution, every patient that comes in essentially with small cell or even other related tumor types, we try to test their DLL3 expression. We're working under the assumption that more DLL3 is better. That has not been proven. This is how we prove it, right? So we look back and we say, all right, are patients that have higher DLL3 expression doing better on tarlatamab on average, or does it not matter? Right? And so we're not necessarily making decisions on the basis of this yet, but this allows us to look retrospectively after we've treated 50, 100, 200 patients with tarlatamab as we have and say, okay, is there a signal there that makes us think that tarlatamab should not be choice number one for every patient, but there should actually be a decision on the basis of their DLL3 expression? If DLL3 is not present, then what do we do, right? So there are other cell surface targets, as I mentioned earlier. I mentioned SEZ6 or SEZ6. I mentioned B7H3. So SEZ6 and DLL3 expression look very similar. So if we look across the subtypes, the same subtypes that are high for DLL3 are high for SEZ6, and the same ones that are low for one, low for the other. So those might not be complementary options. You could potentially double down on the same patient and hit both of them. But if a patient is low for one, they're likely to be low for the other. In contrast, B7H3, this is the target of IDXD, as it's affectionately called. It tends to be sustained or maintained in tumors that are low for DLL3 or SEZ6. And so you could imagine if you knew a patient had 0% expression of DLL3, and if we find that matters, that you may not select tarlatamab. Even if it's best in the total population, you may select a B7H3 ADC. Or if you find that they're ISL1 positive, you may select lurbinectedin using Dr. Shields' data. So this is how we make strides toward ensuring that these patients are not getting one-size-fits-all treatment. And so I just want to end with a comment about where I think the small cell lung cancer treatment landscape is going. So right now, as I mentioned, it's a one-size-fits-all approach, chemotherapy plus immunotherapy, and then this is followed by immunotherapy, potentially with lurbinectedin. In the near future, I think we may see old platinum chemotherapy potentially overthrown by antibody-drug conjugates. The response rates seem higher and potentially more durable. It's crazy to think that we would be treating patients with small cell lung cancer with something other than platinum, but I think it's a real near future potential reality. And I think it's likely that T-cell engagers like tarlatamab will be moved earlier in lines of therapy. It won't be a decision about whether to use them in the second or third line. It will be, do I use them from cycle 1, day 1, or do I use them in the maintenance setting? And I think maybe within the next few years, we will have multiple iterations of this paradigm where we have an ADC and a T-cell engager, and we do testing on patients, and we say, well, based on their subtype and their expression of these surface targets, what is the right ADC T-cell engager combo for this patient that's most likely to work? And that might not be the same for a patient later in the day. And so this is, I think, the hope for all of us, right? That we can have these conversations with small cell lung cancer patients that, as easy as it is for me to have every small cell lung cancer patient that comes into my clinic get the exact same therapy, right? That's just not very satisfying. That's not what any of us want. I would love to have to spend more time counseling patients on their options, more time thinking about their biomarker tests, and more time really doing deep dives into the biology with the patients at my side and talking about why I'm choosing the therapy that I'm choosing than just having a sort of automatic red button that we hit every time a small cell lung cancer patient walks into clinic. And so I will end with that and maybe join Misty over here for any questions that have come up or that she has. split up. How about with my
We play nicely in the sandbox together, so I think we'll be okay. Are we on now? Okay, now we're good. Okay, thanks for bearing with us. Thank you, Dr. Gay. Let's give another round of applause for him. Thank you for. So I just want to take a quick moment while we're waiting for questions in the chat to pop in. Please pop those in as you think of them. But just to highlight here, Dr. Gay has been a huge forever advocate for patients and their loved ones, our smashers impacted by small cell lung cancer. Thank you for everything you've done. Thank you for all the work you've done with longevity. And thank you for helping raise awareness and advancing the field with all of your biomarker research and trials. So thank you. Yes, please. So one thing that I know comes up with our smashers quite a bit is the role of genetic testing or liquid biopsies, NGS, or testing with tissue biopsies. Can you speak a little bit about the role of that? Is it warranted right now in small cell lung cancer, and why does it matter?
Yeah, this is a challenging question. In non-small cell lung cancer cases, this is the default, right? As soon as you meet a new patient with non-small cell lung cancer, you get a liquid biopsy, you get their tissue, you send it for protein testing, mutational testing, fusion testing. I would say in small cell lung cancer right now, it's probably not quite ready for prime time. I think it's always interesting for me as a scientist to have that information, but a lot of those tests are going to yield basically the same results in every small cell lung cancer patient, right? So we're always going to see that TP53 and RB1 mutation if we're thinking about things like liquid biopsies or next generation sequencing of tissue. Now, I think in the very near future, we will be able to answer other questions with these same technologies. We'll be able to use liquid biopsy not only to see which genes are mutated, but which genes are being expressed. So we might be able to tell whether a patient is DLL3 high or low, or ISL1 high or low based on their liquid biopsy. We can certainly do that based on their tissue. I think outside of a clinical trial right now, it probably mostly adds confusion to the mix because I'm not sure there's anything actionable there. That doesn't stop us from trying, right? We're doing DLL3 on every patient, even though we don't know what to do with it, just because we think we might figure it out and might need it a year from now. But I don't necessarily advocate for everybody running back home and telling their docs that they need a liquid biopsy tomorrow and a new biopsy to be sent for subtyping tomorrow. I wish we were there. I would love to hear if you have a contradictory opinion, but I think we're just shy of that point.
Thank you, Dr. Gay. And I think this highlights one of the questions from our last session about, is there a gene for kind of small cell lung cancer? I think that to kind of go back to that question, because I don't think we adequately answer that, my apologies. But I think in certain cases where there might be a family history of lung cancers or small cell lung cancer, I think there is a role for sequencing in that, testing to see if there's certain genes, and then maybe even talking to a genetic counselor if that's something that has been common in your family or in multiple family members. Also, another, I think, avenue that we've always thought about the genetic testing is in patients who have no major risk factors, right? It's unclear kind of why it happened, or it just doesn't, it's in a younger patient. I have patients, as I know Eric does, and you do as well, Dr. Gay, who are in their 40s. I had a patient in their 30s. I know we've had patients in their 20s. Patients in their 50s. We really want to know what is going on. Is there something else that we can act upon, or even help other family members to know and be informed about, or have additional testing? Again, this is case-by-case basis, and really talking to your team if it's right for you. So switching gears, I think, do we have some questions in the chat, Ann?
We do. One really good question is, are there any genetic commonalities between small cell lung cancer and other fast-growing cancers, such as ovarian cancer?
So there certainly are genetic commonalities. So this came up a bit in the last session, which is even between small cell and non-small cell, there are some common factors. So TP53, for example, can be mutated in either of those, and then the the milieu in which the cancer develops ends up making a difference. There are a number of other cancers in the body that fall under the umbrella of neuroendocrine neoplasms, or more specifically neuroendocrine carcinomas. They're almost universally aggressive. Under the microscope, they look a lot like small cell lung cancer, but they can arise from anywhere. They can arise from the skin, the cervix, the ovaries, the gastrointestinal tract. And those often have the same genetics if we do the tests, right? They have TP53 and RB1. So there are definitely some convergent evolution that happens at that level. And then I guess one thing that we haven't directly addressed is the transformation, transform small cell lung cancer, right? And I know that some of the folks that are involved in longevity unfortunately, that's a familiar topic to them. And so these are cases where the genetics start out typical of maybe non-small cell lung cancer, or whatever the tumor is in a particular organ, and then either for reasons we don't understand or because of selective pressure from the therapies, the tumors eventually adopt a more small cell-like phenotype. They look under the microscope more like small cell, they behave more like small cell, they respond more like small cell to therapies. And certainly these patients still fall under a lot of the biomarker headings that we see. You can subtype these cases, you can look for ISL1, DLL3 in these cases and see a lot of commonalities there.
Another question from the audience is, do you feel that there are certain treatments that will make a difference in progression-free survival and durability based on these subtypes? And I think what this question is really asking is there a different pathway? Is there a different first line, second line therapy based on your subtype?
Yeah. I'll answer this first, but I'm clearly biased in this because I'm in the weeds. So I would love to get Dr. Shields' perspective as an objective participant. So I think yes. Obviously I wouldn't be doing it if I didn't think the answer was yes. I think there is already some evidence coming out. We saw a few cases from a group at Dana-Farber at Harvard recently that suggested that tumors that are of the A subtype, this ASCL1 driven group, seem to be more responsive perhaps to tarlatamab, and in response to tarlatamab may shift to one of the other subtypes that's less amenable to tarlatamab. And so I do think that we would get to a point, whether it's with precisely the subtypes that we derived or some variation of those, where we are making that initial treatment decision in a personalized fashion, where we say you are subtype X, and that subtype is more likely to benefit from a DLL3 targeted therapy rather than B7H3 targeted therapy. Or we may get really good at designing therapies so that we can hit all of them at once, right? You can build a better mousetrap so that this doesn't matter. I think in the nearer term, we're probably going to need biomarkers and get to a point where we can select these patients. But Misty, I would love to hear your perspective.
Thank you, Dr. Day. And I agree. I think that the subtypes matter. I was part of one of the original groups that helped find NeuroD many decades ago. And so I do think they matter. And I think that we have to be aware of how the small cell under pressure is going to try to reroute its GPS and escape. And so I think that is where we need a roadmap for every patient, and to know when should we be retesting, or should be retesting, and then how do we kind of detour and set the GPS in a different manner for that patient at that time for the right treatment. I agree. I think if we can smash the cancer in totality and just get rid of all of it, I think engaging the immune system is one way to do that. And how do we kind of flip the switch to help all of our patients, I think is going to be really key here. But thank you for all the efforts of this PRISM study. This is a massive effort. 800, 900 patients will be enrolled. This is a huge effort, but also it will help us move the field forward, because they're doing all this genetic testing, understanding all of these patients' tumors, which has been really not available to date, in terms of the knowledge that we'll gain is going to be huge here. To play a little bit of a devil's advocate, I know that you're saying possibly subtyping is not ready for prime time until this trial results out. If a patient was going to pursue testing with Boston Gene, can you walk us through kind of what would be some logistics, or what does that look like? You mentioned slides. What would a patient potentially ask their team?
Yeah, so it's a what we call a send-out test, meaning that this is not a test that could be done at your home institution. This is a test where Boston Gene would work with the pathology group at your institution, and they would acquire the preferred amount of tissue from either your original biopsy or potentially a new biopsy if you were sufficiently motivated to get this answer. We usually need about 5 to 10 slides worth. These are very, very thin sections, but often with small cell, you get a relatively small amount of tumor initially on the biopsy. So it's not trivial that everyone would have sufficient tissue for this, but we found so far in the PRISM study that most patients do. The tissue would then be sent out, it would be subjected to tests, and then they would report back to your provider, who would then report back to you the results of the subtyping in a pretty easy to interpret schematic like the one that I showed there. And so this can be done. It's billed to your insurance. It's not considered a standard of care, but I think Boston Gene has, in general, agreed that if the insurance gives anybody any fuss, that they will deal with that part of it rather than having the patient deal with it for the foreseeable future.
Another question actually is about the Boston Gene report, and you were alluding to a tumor environment such as the immunotherapy desert. So is that part of the Boston Gene report?
Yeah, that's a great question. And so one of the requirements for doing that test appropriately is that the original biopsy or tissue procurement is done with either a large bore needle, so a wide needle, what we call a core needle, or done via surgical excision. And the reason for that is exactly to your point, Ian, which is that we want to capture not only the tumor cells, but also the environment in which they live. And so when you do a biopsy that way, you retain some of that architecture, and you can see, in addition to the tumor cells, are there immune cells present? Are there blood vessels present? And so the genes that go into the calculation of the subtype do take into account some of the context. And so it's not, the term we use is tumor intrinsic. It's not something that you can derive as accurately if you only have information from the tumor. It helps to have that context.
And what about circulating tumor cells?
Yeah, so I think so small cell lung cancer, unfortunately, has a very high burden of circulating tumor cells and circulating tumor DNA because of its aggressiveness, because it disseminates, because it spreads into the blood vessels early. From a single vial of blood, you can acquire hundreds, thousands of circulating tumor cells. These are live cancer cells that are swimming around in the bloodstream. We can take those, we can put them into a mouse. That mouse will grow a tumor that looks like the patient's tumor. That's really scary to think about. But we can use that to our advantage, right? So we can procure those cells from the bloodstream, from a single vial of blood. And we've seen this recently in another group from Boston. You can potentially profile those circulating tumor cells and get a lot of the same information that you could get from a biopsy. So that group showed that you could tell DLL3 expression status from a simple blood draw using the circulating tumor cells. So I think as these tests advance and we know what we need to be testing for, we're almost certainly going to be able to do all of this, or at least most of it, in blood, either from the DNA in the blood that's shed from the cancer cells or from the circulating tumor cells themselves. Our hope is that with effective therapies, we won't be able to detect any of the ctDNA and CTCs. That'll be a good problem for us to have.
Another question about the subtyping is, if you are not doing particularly well on your current therapy, is there a way, or is there something that you could tell a patient that they should do in order to possibly be subtyped at that point, since it's not really standard of care yet?
Yeah, I mean, I think it would require probably a little bit of legwork on on the patient and their treating physician's part, where they would have to reach out to Boston Gene in order to trigger that. But it certainly could be done. And I think it's a fair question. Like if you're on, say, tarlatamab and not responding to it at all, is it possible that you're one of the patients that fall into the rare P subtype where DLL3 is not expressed and the microenvironment seems quite different and you probably don't respond to a lot of the standard therapies? I think it's a question that I'm certainly curious to know. Scaling that to everybody that's not responding to their current small cell lung cancer therapy is a difficult thing to do, but I don't think it's unreasonable to ask. And if the word gets back to me, I'll make sure that it happens.
And can your subtype change? Is there transformation in small cell, and therefore, as we look forward toward this testing, should we be testing more often at some point? Is that a next step in research?
I think just like we do in, say, patients with EGFR mutations and non-small cell lung cancer, where we survey the landscape periodically, particularly if their tumor starts to grow, to see if something has changed. That's going to be true, but there's some nuance in small cell lung cancer. It's likely not going to be that the mutations change. It's going to be that the subtype or the profile of the gene expression changes. We definitely see evidence of this. Sometimes it's subtle. A tumor that was once ubiquitously one subtype becomes sort of a mixture of multiple subtypes. Sometimes it's complete, and they switch from A to N or N to I. And so surveying that at logical time points, particularly if treatment is not working the way you think it should, I think will eventually be part of the standard of care.
And one last question. I think this goes back a little bit to a question that we answered a little bit in our first session, but has the BRCA gene ever been associated with small cell?
So I can speak on this briefly, and then I'm curious if Misty knows anything that I don't. So certainly there are patients with small cell lung cancer that have deficient DNA damage repair, including mutations in BRCA. That said, it is uncommon to the point of extraordinary rarity that these are germline mutations in BRCA. These are not mutations that folks have been born with or could pass along to their family. They're events that happen spontaneously over the course of life because the DNA is not proofread well in small cell lung cancer cells. And so I am not aware of even case reports of patients that have one of the breast cancer susceptibility programs ending up with small cell lung cancer in a way that was directly tied to it. I don't know if this is something that you have seen.
I would fully echo that. So I think that we, on that genetic testing, and I do test either the tissue or the liquid or both sometimes, we will see these genetic changes, but again, it's in the tumor and not the cells you were born with from mom and dad. And so those are not really the germline or the ones that are inherited, but more so in that cancer cell, a break in the system to kind of to allow more either chaos to occur on the cancer cell or not. And so we do see some of those in our genetic reports as those BRCA mutations, but not from inherited. Now, there are a rare subset of patients who have like a RB or retinoblastoma gene mutation, and sometimes those can be germline or inherited, and those sometimes can have genetic cancer syndromes like retinoblastoma of the eye or cancer of the eye, certain sarcomas, and rarely small cell lung cancer. Again, this is much more rare, but I don't think I've ever seen a report of BRCA germline with small cells. Typically more some of our breast cancers and other female or hormone-driven cancers.
Yeah, during the break, Dr. Singhi and I were talking a bit about Leaf-Raumini in the same way. So this is a genetic syndrome where patients are born with a TP53 mutation, which would put them halfway to a small cell lung cancer if it happened in the right place. But just as you said about the RB mutated patients, these patients often develop other tumor types earlier in their life, and I don't think I've ever seen a small cell case that became of this, often because they're already dealing with leukemias and sarcomas and things early in life, and I think may just not get to that point, sadly.
All right, I think
We don't have any more questions at this time.
wonderful. So I think let's give Dr. Gay another round of applause. Thank you.